Good afternoon, everyone. By my time, it's 4:32. On behalf of South African Association of Botanists, the name is Adeyemi Aremu. I'm the president of SAAB. I would just like to welcome everyone, and we actually appreciate you taking time off to start this year with us on our first webinar. This is the first one for this year, in addition to the SAAB conference that we did in January. So we really appreciate you despite the difficult conditions, load shedding, and every other thing. So at this point, I would like to call on Dr. Anina, who is the chairperson, to please take over. Thank you so much, Anina, and we really appreciate you for taking this on, this very important task. We appreciate your effort. Thanks.
It's a pleasure. Thank you very much. Yes, good. Well, is it afternoon? Good afternoon, everybody. Thank you so much for being here. We are excited to start our new SAAB webinar series for the year. And you can look forward to about 5 or 6 webinars this year from some of our top botanists in South Africa. And tonight we'll start with our SAAB bronze medalist winner. But before I introduce her, I'm just going to list or name a few admin things that we need to get over. Please, during the webinar, please keep your microphone and camera off so that we don't have disturbances throughout the talk. But you are welcome to ask questions at the end. There will be plenty of time for that. So you can raise a hand at the end and ask questions yourself, or you can ask questions through the chat box by typing your questions there, which you can do during the presentation as well. So please feel free to post your questions whenever. And then also, if you do want to get SACNAS points for attending the webinar, please make a note of that in the chat box as well. Just add your name, surname, and SACNAS, and we'll make sure that that gets through the right channels. So please add that in the chat box. And then we are all on different load shedding schedules. So you might experience some lags or delays during the webinar. And it might be something that we can't do anything about. So if you do experience anything, please just wait a minute or two and see if it fixes itself. And if it doesn't, then please let us know and we'll see what we can do from our side. But we'll try to have everything running as smoothly as possible. Yes, I think that's all of the administrative stuff. Then I'm very excited to introduce Mia Momberg. She is an ecologist that's interested in understanding the microclimate and the effect of microclimates on species distributions at different spatial scales. So she did her MSc at the University of Edinburgh in biodiversity and plant taxonomy. And then she continued to do her PhD at the University of Pretoria in the plant sciences department. And currently she's working as a postdoc on the Hot Birds Research Project, where she is, which is based at the University of Pretoria. And here she's investigating the role of mistletoes in providing microclimatic refugia for birds under increasing temperatures. So that also seems like some exciting work. But today, I think we're going to hear more about her very interesting PhD work, which is the reason why she received a bronze medal is because of her great work in her PhD. So we're really looking forward to that. And Mia, you're welcome to unmute yourself and start off. Thank you very much.
Hi, everyone. Thanks very much, Anina, for the introduction, and thanks to all of you for being here. So yes, as Anina mentioned, today I'll be talking more about my PhD's work. The new things that I'm working on aren't quite ready to present yet, but I'll give you a tiny taster at the end of what I'm busy with at the moment. And hopefully the next time I give any kind of presentation, you'll be able to hear a bit more about that. So today I just want to talk about different spatial scales at which wind matters for plants. Okay. So much work has been done on how climatic drivers affect biological diversity and species distribution patterns, including how changes to climates will affect systems. The vast majority of this work, however, has focused on temperature and precipitation. And while we know that both of these variables are really important, it is also necessary for us to consider how other ecophysiologically important variables may affect taxa. So for example, snow cover duration, solar radiation, and soil pH have all recently been found to influence where plant species occur. And this is just an indication of how we need to also consider other variables. Wind is one of these climatic factors, which has very seldom been considered in research. And despite having really clear impacts on plants by firstly causing mechanical damage to leaves and also causing physiological stress. While we do understand the impact of extreme wind events, like for example, hurricanes and tornadoes, those are relatively well documented, we have much less information on how chronic winds or wind that blows kind of every day in a system, how those will affect plant patterns. So this study was conducted in the subantarctic, and this is an ideal study system when wanting to work on wind, because it's a chronically windy system with wind on Marion Island, where the study was based in particular, blowing on most days of the year. And there's also very low levels of human disturbance, which allows us to kind of disentangle the effects of climate and other factors a little bit better than we would have been able to do if there were a lot of anthropogenic disturbances. So I'm going to start off at a fine scale, and then I will kind of zoom out to a broader scale later in the talk. So at a fine scale, to investigate how wind affects plants, we focused on one particular part of the island, which is highlighted in this little red block on the northeastern side. And in this area, we laid out grids. So there were 9 study grids, and they were laid out to maximize topographic heterogeneity. So if you have a look on the picture on the top left, you can see that not all of the grids are laid out in exactly the same way. They are kind of randomly spaced, and that's just to capture as much variation as possible. So these 9 grids, each grid was 8 by 20 meters in size and was then divided up into 160 cells of 1 by 1 meter in size each. So this then across the 9 grids results in 1,440 quadrats. And within each of these quadrats, all the vascular plant species were identified to species level, and their cover was then visually assessed. Cover of bryophytes was also recorded, and then a whole range of abiotic variables were measured, including soil moisture, soil temperature, slope, aspect, and rock cover. So with all of this data, we have a really, really nice idea of what's happening within each of these cells and all the different aspects that are taking place. So after that, we obtained drone imagery for each of these grids, which I was really grateful for, because this was a bit of a challenge given the wind conditions on Marion. So the last grids were actually flown on the very last day that we were out in the field before we had to return to the mainland. So it worked out just in time. And this drone imagery allowed us to create a digital surface model. So a digital surface model allows you to see the height differences within the image. So almost like an elevation model. And so the nice thing that we've had in addition to this project, which has really been key to us being able to do this work, is that there's been a collaboration between the plant science department at the University of Pretoria and the Department of Mechanical and Aeronautical Engineering. And so they've done a really fancy computational fluid dynamics model. And we had 17 anemometers set up across Marion Island over a period of 2 years, which we were able to use as reference data for wind speed, wind direction, and wind turbulence. And so using the computational fluid dynamics model and then using the values from the island to kind of see whether it was really performing well enough, we now have a really nice wind map of the island. And so I could extract the wind speed from that map. So the idea here was to get an idea of the wind conditions experienced in each cell. So using that digital surface model from the drone imagery and the dominant wind direction, I was able to calculate first the wind exposure that was present in each cell, and then I multiplied that with wind speed to create this unique wind stress metric, which would give us an idea of the wind stress that plants were under in each particular 1 by 1 meter quadrant. So I just wanted to give you a little bit of a visual idea of what the grids look like. So these are only 4 of the 9 grids, but you can see on these pictures that each of these grids looks quite different. The vegetation looks quite different, the species that are occurring there look different, and we have a very big difference in the amount of vegetation cover that's present. And so indeed, when we look at species richness, for example, we do see really big differences between the different grids, but then within each grid, there are also differences where sometimes even 2 cells which are next to each other may have very different species richness values. So generalized additive models and generalized linear models were then used to model species richness, vegetation cover, and the individual species occurrences and cover. And then after that, to look at species composition, we used non-metric multidimensional scaling. And to determine whether there was nestedness, we also did a nestedness analysis to see whether species were nested along the wind stress gradient. So I'll start off with species richness. So for species richness, including wind, produced a better model. So for all of these models, first a model was run that did not include wind as a predictor, and the model was then run a second model using all of the same predictors from the first model, but also adding wind stress to compare whether the addition of that variable actually produced a better model. So for species richness, it did indeed produce a better model, and wind was in the top 5 most important predictors. And not only was it in the top 5, but a really, really cool result was that it was the most important predictor in predicting species richness at these fine scales. And so I just want to point out on this figure that temperature, we have 2 temperature values, one for winter and then one for summer. And then we also have soil moisture on here. These are the variables that we traditionally use when we are thinking about climate. And although they are important and they were significant, it's just really insightful to see that in some systems, wind might in fact be driving patterns more strongly than these temperature and moisture variables. So moving over to species cover, once again here, the model which included wind stress as a predictor performed better than the one without it. And here wind gets bumped down 1 notch, but it still is the second most important predictor. So in this case, we see that rock cover was the most important predictor of species cover. And that makes sense because in a really high rock cover, so we do have some areas in these grids that are complete rock cover, and those areas would just preclude species from growing there because there's not really available substrate for them to grow. So that's not a surprise. And then we have wind stress second and the summer temperature coming in third. So this could also be on the wind stress graph. If you have a look, you'll see that in the tail end, actually under high wind stress conditions, the vegetation cover is slightly higher. And this was a little bit counterintuitive, but this could be due to the fact that bryophytes are really important in the system. In fact, we have way more species of bryophytes here than vascular plants. And under strong wind conditions, they might not be able to survive the desiccation. And this might then allow some extra space to open up for vascular plants to actually achieve greater cover. Then we can dive into species composition. And again, here, wind is a significant driver. It's a little bit less important than for the species richness and species cover analyses. It comes out as the fourth most important predictor. We see really low nestedness, which means that there's turnover along the wind stress gradient. So as wind stress changes, we have species replacing one another under these different wind stress conditions, while species show different responses, and that makes complete sense. So when we look into the individual species, we now know that they are replacing each other along this gradient. So we expect them to show different responses. And what was really interesting is that out of the 16 species that occurred in these grids, 12 of those species occurrence patterns were significantly affected by wind. So on this graph here, I've plotted the significant relationships in solid lines. So all of these indicated by the dashed lines are those that were not significantly influenced by wind. And you can see that most of those were ferns. So the first panel of the graph, we actually had 3 ferns that weren't significantly affected by wind, and then there was 1 grass. And so most of these species, when we really look into the species life history traits and where these individual species grow in their characteristic environments, these patterns come out quite nicely. So I'm just going to give you 2 examples. So firstly, for example, Phlegmarius saururus, which is a club moss, has recently undergone a taxonomic revision. It used to be lycopodium. It's indicated by this dashed orange line. And you can see that firstly, the relationship was not significant with wind, but also that that line is nearly horizontal. This was unexpected at first, but then the more I looked into the species traits, it's likely driven by the fact that this species is most often found growing in rock cracks. So like this photo here, they really are almost always found growing in rock cracks. And here the roots will be more protected from desiccation, providing like a little pocket where they are more protected from wind. And for that reason, maybe not, we aren't seeing that their occurrence patterns are being driven by the wind conditions. And then on the other hand, we have Ranunculus bitornatus, and this is the Antarctic buttercup. And so it was significantly affected by wind stress, with its probability of occurrence decreasing under really very high wind stress conditions. So this species is characteristic of wet mire environments. We don't find it really outside of these mire habitats. And when wind stress becomes too high, it's likely that these habitats and the species growing there may dry out. And that's why we do see a slight decrease in its probability of occurrence under higher wind stress conditions. So for the fine scale species patterns, it's clear that wind affects where species grow, where individual species grow. It affects species richness, it affects species cover. So all these fine scale community parameters are affected by wind. Question is, where else may wind be important in driving individual species occurrence patterns outside of Marion Island? So the most obvious would be that in systems with chronic wind, so other systems, often coastal systems or montane systems, where we also see that there is wind on many days of the year and that strong winds occur frequently, those systems are also likely to be really affected by wind. But we also actually might expect wind to be important in some other areas as well. So for example, areas that have a lot of loose material available for aeolian transport. So this would be, for example, dunes or deserts. These areas are likely also going to be affected strongly by wind, because this loose sand or other material can easily be picked up and used as mechanical or involved in mechanical damage when it's blown against plant parts and causes abrasion. And also there could be a really big redistribution of the substrate. So you would imagine that when substrate's not stable and the substrate material is moved around often, it might be more difficult for plants to establish and grow. And then the two other areas that wind may be really important is areas that are already dry or that have really thin soils, as the wind will just desiccate these areas a lot quicker than areas that receive more moisture or that have deeper soils that are able to hold more moisture. So it's clear that responses to wind are species and site specific. So how species and communities will respond to changes in wind conditions can also be confounded by plant-plant interactions. And then we have the added complication that changing wind conditions in conjunction with changing precipitation regimes and temperatures may also lead to areas that are now, either now or in the future, going to experience novel climates. So these would be climates that species have not yet experienced in these areas, and how species will respond to these new conditions remains to be seen. It's also clear that there are interactions between microclimate and topography, and so this may also be important in mediating responses. So, for example, even if wind conditions or wind speeds increase, there might still be certain topographic areas that produce microclimates similar to what we see today. So maybe if there's a depression, there will be more moisture and will be more protected from wind speeds. So it's also really important to consider how these interact with each other. So this is kind of like the baseline that we started off with at a fine scale. And after seeing that wind really does have an effect on these community parameters, we wanted to zoom out and take a broader approach to see whether it also affects plants at broader scales. So moving to this broader scale, we then looked at the total vegetation distribution across the entire island. So to start off, I obtained satellite data with a resolution of 30 meters from the advanced land imager. And this satellite data was then used to calculate the normalized difference vegetation index, as well as the soil adjusted vegetation index. So I'm sure that many of you are familiar with NDVI. It's something that we use often. And so SAVI is a very similar metric to NDVI. It just minimizes soil brightness by using a soil correction factor. So this is useful in areas where you might have low levels of vegetation cover and there might be large areas with bare soil. You don't want that reflectance to affect your calculations for NDVI. So I ran these analyses on both of these metrics, but there actually turned out to be no difference between the results for the two. So I'm going to only present those from the NDVI. And so for the predictors, I used ArcGIS to calculate the terrain ruggedness, and that was done using the digital elevation model for Marion Island, as well as the curvature, the geology, the particular geology type, the potential direct incident radiation, and then the wind speed and the wind turbulence values. Those were obtained from the same CFD model that I mentioned to you for the fine scale work. So what we did was to generate, also in ArcGIS, 10,000 random points that were just scattered across the island. So there were some criteria here. Points were not allowed to be in lakes. Points were not allowed to be on geology types that were scoria. This was done because we know that on the scoria cones, there's very, very little vegetation growth. So we didn't want to include that because those values wouldn't really be useful to us. And then there were also buffers included that points weren't allowed to be too close to one another. And these 10,000 random points, all of the predictors I've just mentioned were then extracted for each of these points. So I used the generalized additive model again. And the very first thing that came out was that elevation was super, super important. So this is not a surprise to us. There's a really strong temperature gradient with elevation on Marion. So along the coastal areas, it's much warmer. And then as you move towards the interior of the island, there's a sharp increase in elevation, and that's when it becomes much colder. So because elevation had 52% variable importance, it was really difficult to see the effects of the other predictors. So what we decided to do was to run a model that first included only elevation as a predictor, and to then take the residuals from this model and use those residuals as the response against all of the remaining predictors, which would allow us to see the effect of all of these predictors after already accounting for elevation. So these are the results from that. And they are all, the graphs are ordered in order of variable importance. And so you'll see that potential solar radiation played the largest role in determining vegetation cover after accounting for elevation. And that can be related to multiple things. It could be surface temperatures, it could be evaporative demand, soil moisture, light availability for photosynthesis. So this, of course, is not an absolute measure of solar radiation because it cannot account for cloud cover, but it just reflects the potential solar radiation that an area can receive based on the latitude and the slope of that particular cell. And then after that, we see that there's lower vegetation cover than expected based on elevation for areas with high terrain ruggedness. And this again likely reflects high rock cover, which is just limiting the vegetation growth. And then in the third panel, you'll see that once again, wind speed is a significant predictor. It's not the most clear relationship, and it's not got a very high importance relative to the other two, but it still does come out as a significant predictor. But wind turbulence, which is the bottom middle panel, that is not a significant predictor of the total vegetation cover across the island. So to stay at this broad spatial scale, but to maybe get a bit of an understanding of things at a different level, we also wanted to look at the distribution of individual vegetation types and whether not only the total amount of vegetation is affected, but these different types occurrences are affected by wind. So to determine if the distribution of these vegetation types across the island was driven by wind, 24 transects were sampled, and these transects were scattered across the island, and they ran from sea level towards the interior. So on these transects, plots of 3 by 3 meters in size were sampled every 100 meters, or if before you reach the 100-meter mark, there was a really clear visual change in vegetation, that area would be sampled as well. So after these transects were sampled, we added another additional 17 plots at high altitudes. And these were just added to make the data set more representative of all the different altitudinal categories so that we didn't have a bias towards not having enough data from higher altitudes. And then within these plots was a similar kind of method to the fine scale approach. So all the vascular plants were identified, the cover was identified, the bryophyte cover was recorded, and that was done in different bryophyte categories. And for these, then that was all that was recorded. There was no measuring of abiotic variables in these quadrants like at the fine scales. So using this plot data, which had all the vascular plant information and the bryophyte information, we could then use the existing key to vegetation types, which was created by Smith and Steenkamp in 2001 for Marion Island. And then each quadrant was then assigned a particular vegetation type based on this key. The same predictors were then used for the NDV as for the NDVI analysis, which I've just shown you. And then here we can see the relationships between each of the 6 vegetation types. So each vegetation type has its own color, and then the 5 predictors. And so you'll see that for some of these panels, there are more lines than others. So only significant relationships were plotted to make it easier to kind of read these graphs. So you can see in the very first graph that for elevation, it was a significant predictor for the occurrence of all 6 of the vegetation types. And then the graph below it is for wind velocity. And for wind velocity, it was an important a significant predictor in determining the occurrence of 5 out of the 6 vegetation types. So the only one that's excluded on this wind velocity graph is the biotically influenced vegetation. And then for the remaining 3 predictors, you see that they only significantly explain 2 or less of the vegetation types occurrences. So to just dive a little bit into some of these patterns, we can look first at fell field. So you'll see that that's the dark blue line. And you can see that it has a really strong relationship with wind velocity. So there's a larger probability of fell field vegetation type occurring under higher wind speeds. And so fell field vegetation typically occurs on exposed ridges and plateaus where topography causes the wind speeds to be higher in comparison to the adjacent low-lying areas. And so that's why we see this pattern here. And then in contrast, you can see that the mire environments, which are in orange, and the slope and fern break environments, which are in green, those were more likely to occur at lower wind speeds. And so, as I mentioned when talking about the Antarctic buttercup, mire habitats are typified by wet soils, and the desiccating effect of wind may limit these communities from establishing under high wind speeds. And then fern break was also that's the green line as a reminder, was most common under the low velocities. And that's possibly due to ferns being very sensitive to drought stress. So there's been previous work done to show that only small changes in leaf water potential can lead to permanent leaf death in ferns, instead of the stomata just closing as we would see in many other species. So we can actually disentangle a lot of these effects and understand why we see these patterns. So just wanted to also highlight that for these different vegetation types, not all of them were as well explained by the models that we ran. So we had some of the vegetation types like the coastal salt spray and the polar desert that had the high deviance explained, which indicates to us that the model is performing well. But there were others like the mire environments that had really low deviance explained percentages. And this just likely indicates that there are other variables at play that we have not considered, that haven't been included in these models. So there is still some work to be done in understanding exactly why all these vegetation types occur where they do. And then the columns that follow in the table, those show the variable importance values as a percentage for each vegetation type. So you can just see that elevation and wind velocity, those two were the 2 predictors that overall across all of the vegetation types had the highest variable importance values. And again, that's relating to temperature in terms of the elevation and also the wind velocity. So for example, the coastal salt spray communities, those were really well explained by this model, where they are found. And they are found further inland on the north and on the west coasts than on the south and east coast. And this is just showing how the dominant westerly and northwesterly winds in the Southern Ocean will then carry salt from the ocean further inland on these sides of the island, which then allows this vegetation type to spread further inland than it would on the sides on the south and the east. So the final step for this broad scale approach is to now redo the analysis. So this is a really large data set, which I think is amazing. I'm very proud of the fact that we have such a large data set, and I think it allows us to really confidently extract patterns. But due to the large size, we have significant spatial autocorrelation. And while generalized additive models do deal with some spatial autocorrelation, we just want to make sure that these results will remain the same when we explicitly account for spatial autocorrelation. So I'm going to be rerunning the broad scale analyses using spatial lag models, spatial lagged X models, and spatial error models. And hopefully we will see similar patterns, or even if we don't, we'll then be able to conclude this and hopefully publish this. So wind velocity, to conclude, we've seen across spatial scales on Marion Island is a significant predictor at fine scales for individual species and communities, as well as at broad scales for vegetation types and overall vegetation cover. We see that under high wind speeds, we have lower plant densities. And then something that I think has also been an important outcome from this work is that wind turbulence is not as important. For most of the analyses, wind turbulence was not a significant predictor. Unfortunately, wind turbulence is much more difficult to measure. You need a lot fancier equipment, and you need more consistent measurements. And so this is a nice thing that we can take from this, that in other systems, when you are doing studies, you should rather focus on measuring wind speed and wind exposure. And luckily for all of us, those are the two variables that are much, much easier to measure. So we don't need to spend too much resources on trying to measure turbulence because it doesn't seem to be that important. Like I've already mentioned, the different vegetation types are affected by wind. So salt spray, when wind conditions are rough, is able to spread further inland. Foul field is limited to these wind-exposed ridges across the island. When wind speeds are low, we have mire and fernbreak communities. And this really then asks the question that under changing wind conditions, where both wind speeds and wind directions may change, what will this have in store for where these individual vegetation types are able to occur? So we do know that a lot of changes in wind conditions have occurred globally, but the biggest changes have been in the Southern Ocean. And so that's, of course, also where Marion Island is situated. So I think it's also really important that we now have a baseline understanding of what is happening on the island. So perhaps in 10 or 20 years, we'll be able to do a comparison of how things have changed given the changes in wind conditions. Of course, we always want to know where else our studies may be applicable. And so in systems like Marion, where we have snow, wind may also redistribute the snow. And so that could have many implications for species where areas that are then covered in deeper snow will be more buffered from temperature extremes, and those areas where snow has been removed by wind will be affected by more freeze-thaw cycles. So for example, in the Scottish Highlands, we have many snowbed communities, and those communities have specific species that are only able to survive the winter by being in these snowbeds where temperatures are really buffered. So if the snow is removed, they will not survive the winter. So there may be implications in habitats experiencing snow. And then in systems where we have fires, which is of course not on Marion, but elsewhere, wind affects the temperature and the strength of the fire, having large implications for whether the fire actually then ends up being beneficial or negative. And then once again, like I mentioned before, in dune systems where substrates can be moved around a lot. So wind conditions are related to multiple levels of biological communities, and really should be stressed that it should be considered as a climatic driver in future. Temperature and precipitation are important, and I think we need to continue looking at those, but we need to also start thinking about what else we can add. I also just wanted to show this picture, which was from my field notebook, that there may be some other effects that we haven't yet looked at. So intra-specific plant traits, for example, may be affected by wind conditions. So this is the same species, which I found growing maybe 5 meters apart, these two plants. And the one was growing in like a little rock crack that was protected from the wind. And you can see that the plant's much taller than the one that was wind exposed. So we may also want to consider other measures of plant performance. And then it's just possible as well that plants, we know that plants and animals may struggle to adapt fast enough to changing conditions. We've seen this with temperature and precipitation, so they may also struggle to adapt fast enough to changing wind conditions. And then wind conditions will also interact with temperature and precipitation regimes. So for example, regions that experience reduced precipitation will maybe be disproportionately affected if they also experience increased wind speeds. Or in contrast, places that might experience increases in temperature, these increases could be mediated by cooling winds. So these interactions also need to be investigated. And then I've already mentioned to you that the wind turbulence was not as important, and so we need to focus on wind speed and wind exposure. So this is kind of the end of this work, and then I promised a very small taster of what I'm doing now. So I've moved to a very different system. Marion was cold and wet and windy, and I've now been working on a postdoc with a hot birds research project, and this has been based in the Lowveld. So we are looking at mistletoes, and we are installing small weather stations within the mistletoes and within non-host trees that don't have mistletoes in them, containing a Stevenson screen measuring air temperature and humidity, and also a black bulb measuring a proxy for bird operative temperature. And the idea here is we want to see whether mistletoes are providing climatic refugia where birds may find respite from really hot conditions. So yeah, if this interests you, keep your eyes peeled. Hopefully something will come out of this project soon. And I just really want to thank all of you for making the effort to attend today, and really big thanks to all of my co-authors that were involved in my PhD for making it all possible and a really great project, and also to the NRF for funding. Yeah, I'm really happy to answer questions now, and if you think of something later, you're also welcome to pop me an email. Thank you.
Thank you very much, Mia. That was great. I really enjoyed that, and I'm sure everybody else did as well. So yeah, everybody, you're welcome to ask questions. So you can use the chat box function if you want, or you can raise your hand and I'll try to find that. And then you can ask your question if you have any. Let me just double check here. Don't see any questions yet, but I do have a question. So when you're talking about wind, yeah, I don't know much about it, but in our system here in the Southern Cape, the wind is very important in shaping where the forest pockets occur in between the fynbos, because the wind determines where the fires move. So I was wondering, so wind direction, prevailing wind direction is really important in this area. And I was wondering if there are already predictions about if that is something that's possible to change with climate change, the prevailing wind directions, and how drastic that would be, if you know anything about that.
Thanks, Anina. Yeah, so I'm not sure in particular for the Southern Cape, but globally we do already see shifts in wind direction. So for example, in the whole Southern Ocean, there's this westerly wind belt and it's shifting. So that then affects the prevailing wind direction. And globally, there have already been shifts. So there aren't that many predictions for what the magnitude of these shifts are going to be. There are predictions that the shifts are going to continue, also because of the differences in temperature from the ocean surface, which then also affects the winds. So I think that there definitely may be changes to the predominant or prevailing wind conditions, but and the direction, but I just don't know how bad it's going to be.
Yes, I can imagine. That's always going to be the big mystery. How bad is it going to be? Well, yeah, thanks for that. That's really interesting and a bit scary. I'm just going to give some more time for questions to come up. Sometimes it takes a while. And if anybody else sees a hand up that I missed, Dave Berger, I see your hand up. Oh, there's another one. Okay, Nishi first, and then Dave. Thank you.
Mia, thanks for a great talk. I'm glad to hear what you've been doing. I have in terms of the fern distribution, it seems like 15% of the distribution was determined by the geology. So I was wondering what the geologic differences are. And in fact, if you did find kind of species that were more restricted to one type of geology over another, I mean, 15% seems pretty high on your setting.
Thanks, Nishi. So yeah, we didn't see any fern species that were restricted to only one particular geology type. But the main differences on Marion in geology are that there are pre-glacial and post-glacial lava flows. So the volcanic lava that was deposited pre the last glacial maximum, that's been smoothed by the glaciation process, where the post-glacial flows are really rugged. And so it's just a very big difference in the I guess ruggedness is the right word of these two geologies. And so I imagine that perhaps on the more rugged post-glacial flows, ferns might be able to attach better to that kind of substrate. I don't know. Yeah, I haven't actually investigated that further.
So you're referring to kind of the surface, the surface texture of the rock might be contributing to the colonization and the attachment of the fern rhizoids. In the same, I mean, why weren't the bryophytes included in that analysis? I wondered whether the bryophytes were responding more to the geology, especially to the surface features, than also.
So the bryophytes, our biggest issue with the bryophytes is that nobody has worked on bryophytes on Marion Island for decades, and so there's nobody who's actually able to identify them reliably. So I would have loved to include the bryophytes, but there's so many, and I'm pretty sure there are undescribed species, and even those species that are described, it's really difficult to identify them, and there's no one else really at the moment who can help with that.
Thank you.
Thank you very much. Dave, will you please go ahead and ask your question?
Okay, thanks very much. Can you hear me all right? Sorry,
Yes,
my
Prof.
camera's not working. So congrats, Mia, on a very nice study and getting the SAAB medal. That's really great. Lovely talk. So I just got a couple of questions. The first one is, when you have an environment where you don't get a very, very strong prevailing wind from one direction, is this study very much more difficult to do? This type of study?
Yeah, I would imagine it is more difficult because, I mean, when you have wind coming from only one direction, it's much easier to determine the exposure of a particular cell. So if maybe a large boulder or a ridge is blocking the wind from that prevailing direction, you would know that there's a leeward and a windward side. When that's not the case, it would be much more difficult. But I think that you would be able to, if you had the data, on how often wind is coming from each direction, you might be able to scale that wind exposure by frequency from each direction to kind of get that into one metric, but I don't know if it would give you as clear an answer.
Right, great. Now that makes sense. And what about the impact of the wind on the distribution of alien plants, and maybe in terms of reproductive traits? Have you got any ideas on whether that is an effect there on Marion Island?
So I don't know if it has an effect, but I think that it's very likely that it will have an effect. So a lot of the species there are wind distributed. For example, one of the predominant alien species is a grass. And of course, the grass seeds will be dispersed by wind. So I think there is a chance that changing wind conditions might affect where we find alien species, but equally it could also allow native species to spread to areas that they haven't been able to colonize before. So I guess it just depends on how well the alien species is able to outcompete the natives.
So there weren't any particular alien species which were dominating in certain environments?
No, not in any of the ones that we've looked. I mean, we did include alien species in the study. So where alien species were found in the fine scale grids and in the transect data, we kept those species in there, but we didn't have very high numbers of alien species. And so obviously there are some areas that have a lot more alien species. So around the base, there are a lot of aliens. So maybe it would be nice to do the study where you focus the data collection on an area that was heavily impacted by aliens to determine the effects, because the areas that we looked at did have some alien species, but just not a very high frequency of alien species, which is good.
Great. Okay, thanks very much, Mia, and best of luck with your postdoc.
Thank you.
Thank you very much, Mia. I'm looking for other hands. I don't see any hands up right now, but there's an interesting or comment from Eugene Moll in the comment section or chat box, where he says that the Cape of Good Hope Nature Reserve would be a very interesting study site. I don't know how familiar you are with that area, but it can be very windy there. How some fynbos heathland communities survive the continuous southeasterly winds would be very interesting, not just the ecophysiological adjustments, but the pollination and the seed dispersal as well.
I agree, that would be really, really cool to see. And also to see whether some pollinators maybe aren't able to tolerate certain wind conditions. I know there's been some work on Marion done about when some pollinators come out in relation to the weather.
Yeah, my one supervisor's theory about sugarbirds' very, very sharp claws is that they've got that to hold on to the approacher flowers in the wind. I see, is it Peter answered or made a comment to Dave Berger as well. Another Marion Bay student looked at wind dispersal of the island's vascular plants and didn't find a difference between the aliens and the native species. So that's interesting. All right, let me just quickly scan for any hands that I might have missed. Right. Don't see anything. All right, are there any other questions from the audience? Don't want to miss anybody. There's one, is it Eugene? Please go ahead.
Can you hear me?
Yes.
Can you hear me now? Hello?
Yes. Yeah.
Okay. Going back to Cape Point, what's interesting to someone who grew up in the savannas and came down here to the Cape is that a lot of Cape species are monoecious. So all the restios, for example, which dominate vast tracts of Cape of Good Hope Nature Reserve, are monoecious, and also they're generally wind pollinators as far as I'm aware. So that puts some very interesting challenges to the growth and health of those communities. And to me, it's something that really needs to be looked at carefully, because we don't really understand a lot of the dynamics of some of those communities. I don't think anyway. So let's hope that someone will get onto it one of these days. Mia, more money, another research project, please.
Yeah, if I can gather enough courage to learn the fynbos species, that sounds like a really, really good idea and very interesting project.
Yes, definitely. We'll need your expertise there. Just get a student that can ID the plants. Sorry, Eugene, do you have something to add?
Yeah, just to say, I came from KZN to UCT many years ago to a totally alien flora. I immediately ran into the Afromontane forest because they were at least something I could recognize there. And it's not that difficult to learn some of the Cape species, particularly if you put them into physiognomic growth form habit types. So instead of being so focused on the species, look at growth forms and how do different growth forms, and then you can bundle, the 1,000 species into 20 or 30 growth form types, so you can, simplify it in that way. I don't think that what happens in the Cape to me is that people come to the Cape and get so embroiled in species and their species conservation that they lose sight of the bigger picture. And I think that's a real problem. Anyway, that's just my opinion.
Thank you.
Thank you very much for the comment. Are there any other questions? Don't see any hands or any comments. So I think that is it. Thank you very much, Mia. This was really great. We appreciate your time in preparing this and everything, and it was really interesting. And yeah, we do look forward to your new research as well. So that seems very exciting. So I hope everything goes well and that all the plants and birds and machinery works together to make things a success. And thank you very much to the audience for being here. I hope this was great. This recording of the webinar will also be available on the SAAB website. So you're welcome to access it from there as well. Yeah, any last words, Mia?
No, just thanks for inviting me, and I hope you enjoyed it, and thanks to everyone for coming to listen.
Great. Thank you very much.
Bye.
Thanks. Adiyemi, anything else, or can we close?
No, thank you so much, Janina, and thanks to everyone. I think this is just the beginning. We have many more coming.
Yes.
So nothing more from me. I just want to thank everybody again. Thank you so much. And
Great.
Thank you to Elizabeth for organizing this. Thank you so much. Bye.
Yes, our next webinar will be in May. So we will send out the advert and details about that. But thank you, everybody. Have a good night.